All right, welcome. Today, we are going on a journey into a world that is literally all around us and even inside of us. And yet, for almost all of human history, it was completely invisible. We are diving into the history of microbiology. I mean, just think about that for a second. For thousands of years, empires rose and fell. Devastating plagues swept across the globe. Bread would rise, wine would ferment, and it was all being driven by these tiny, powerful forces that nobody could even see. This invisible world was shaping our reality, and we didn't have a clue it was even there. So let's pull back that curtain and see how we finally discovered it. So, what exactly is microbiology? Well, it's the science of that invisible world. It's the study of all that microscopic life. We're talking bacteria, fungi, viruses, and all these other tiny organisms you can only see with a microscope. Okay, so our story starts way before we could actually see any of these little critters. Before microscopes, all we had were, well, ideas. Theories about what was causing disease and making food rot, mostly born from just observing the world and, a bit of clever guesswork. Yeah, the path to actually seeing this world was a long one. It really started with just speculation, like from Marcus Varro back in ancient Rome, who had this wild idea about invisible animals causing disease. Then, fast forward hundreds and hundreds of years to 1665, and you have Robert Hooke using an early microscope to see the first cells in a piece of cork. But the real game changer, that came in 1674, when a guy named Anton von Leeuwenhoek became the first person ever to see living microbes. And this is what he saw. It was all possible because of this incredible leap in technology. And what's amazing is, Leeuwenhoek wasn't some highly trained scientist. He was a Dutch cloth merchant who just got really good at grinding lenses. His simple little microscopes were so powerful, they basically opened up a portal to a hidden universe in a single drop of pond water. He saw all these tiny things swimming and zipping around, and he called them animalcules, little animals. For this, we now call him the father of microbiology. Okay, so seeing these animalcules was one thing, but it opened up a huge, huge question. Where did they come from? Did they just, poof, appear out of nowhere? This question kicked off one of the biggest scientific fights of the time, a debate that would rage on for more than 100 years. So you had two camps. In one corner, you had the theory of spontaneous generation. This was an old idea that people had believed for centuries, that life could just spring into being from non-living things. Like how you see maggots on rotting meat. They just thought the meat made the maggots. And in the other corner, you had this new idea called biogenesis, the principle that life can only come from other pre-existing life. A pretty straightforward disagreement, but a really profound one. This is where Louis Pasteur comes in with what might be one of the most beautiful experiments in the history of science. First, he took some nutrient broth and put it in this flask with a long, curved S-shaped neck. Then, he boiled the broth to kill anything that might be living in it. Now, here's the genius part. The S-bend let fresh air get in, which was important, but it trapped any dust or microbes from the air in the curve. And guess what? The broth stayed totally clear, totally sterile. But the second he tilted the flask and let that dust touch the broth, boom, life appeared. And just like that, Pasteur proved it. The microbes didn't just appear out of thin air. Nope, they were just hitching a ride on those tiny dust particles. He proved, once and for all, the principle of biogenesis. Life comes from life. And that discovery, that was like the starting pistol for the entire modern era of microbiology. With that whole spontaneous generation debate finally put to bed, the floodgates of discovery just burst wide open. Scientists now knew for a fact that microbes were real, they came from other microbes, and they were absolutely everywhere. The race was on to figure out, what are these things actually doing? This next chunk of time, roughly from 1857 to 1914, we call the golden age of microbiology. I mean, it was less than 60 years, but the progress they made was just mind-blowing. Our entire view of disease, of food, of life itself was completely rewritten in just a few decades. And Pasteur, man, he was right in the middle of it all. He figured out that microbes are what cause fermentation, the very thing that gives us wine and beer. And then he invented pasteurization to stop them from spoiling things, a process we still use on our milk today. But maybe his biggest legacy was championing the germ theory of disease, this totally revolutionary idea that invisible germs are what make us sick. And that idea, that led directly to the first modern vaccines. At the exact same time, over in Germany, this guy Robert Koch was busy building the toolbox from microbiology. He was the one who figured out how to grow bacteria in pure cultures, so he could isolate just one type of microbe at a time. This allowed him to pinpoint the specific culprits behind deadly diseases like anthrax and tuberculosis. He even came up with a set of rules, which we call Koch's postulates, that act like a blueprint for proving that a specific microbe causes a specific disease. It's a method that scientists still use today. So all these pioneers during the Golden Age were finding this whole zoo of tiny creatures. But on a really basic, fundamental level, what were they? It took a while, but modern biology eventually figured out that all these microbes and, in fact, all life on Earth is built on one of two basic designs. Right, so Leeuwenhoek's animalcules were just this chaotic mess of different shapes and sizes. But as our scientific tools got better and better, we started to see the patterns underneath it all. We realized that this massive diversity of tiny life could pretty much all be sorted into two major buckets based on how they're built inside. First up, we've got the prokaryotes. The name literally means before the nucleus. These are the simple guys. They're like a one-room studio apartment. Their DNA just kind of floats around inside in a region we call the nucleoid, and they don't have any of the complex little rooms or organelles that other cells have. This group includes all the bacteria and archaea. And then you have the eukaryotes. Their name means true nucleus. These are the complex cells. Think of them like a big mansion with lots of specialized rooms. Their DNA is tucked away neatly inside a nucleus. And they've got a whole bunch of these specialized organelles doing different jobs, like mitochondria making energy. This group includes a huge range of life, from single-celled protozoa all the way up to plants, fungi, and us animals. Let's just put them side by side for a second. The biggest, most important difference is right there at the top, the nucleus. Eukaryotes have one, prokaryotes don't. That's the main thing. But you can also see other big differences, like how their DNA is shaped, and the fact that eukaryotes have all those organelles while prokaryotes don't. This basic difference in structure leads to big differences in everything: their size, their complexity, even how they make copies of themselves. You can think of prokaryotes as the ultimate survivalists. They have this simple, stripped-down, super efficient design. It's a design that's worked for them for over 3.5 billion years, letting them live in every single nook and cranny of this planet. They are the true originals of life on Earth. Eukaryotes, on the other hand, went a totally different route. They went for complexity. By building all those internal compartments, those organelles, they could have specialized departments within the cell, each with its own job. This division of labor is what allowed them to get much bigger and eventually to team up and create complex multicellular life. You know, things like us. So, after all that history and all that biology, why does it matter? Well, because understanding this invisible world has completely transformed our world. From the germ theory that gave us modern medicine, life-saving vaccines, and antibiotics, to the microbes that help grow our food and even clean up oil spills, the impact of microbiology on our daily lives is just staggering. It's everywhere. So we've gone from not even knowing these things existed to being able to read their genetic code. We've learned the fundamental rules of this invisible world, which leaves us with a really exciting question for the future. Now that we know the rules, what are we going to build with them? From engineering bacteria to make new medicines, to designing synthetic life forms from scratch, the next chapter in this incredible story is really just getting started.